Childhood poverty is a major public health problem. As an estimate of the number of children affected, there are nearly 15 million children in the United States who are currently living in households with incomes below the federal poverty line, and another 15 million in other industrialized nations who live in relative poverty. This represents more than 20% of children in the United States, and a range across industrialized countries from 4.5% Iceland to 25.5% Romania. While these official rates are staggering, they underestimate the number of children affected by poverty worldwide, as published measurements often rely on outdated assumptions about family expenditures and resources. The full burden of poverty for children frequently includes early and repeated exposure to stress and environmental hazards. Empirical investigations have noted children living in poverty are exposed to more family turmoil, violence, separation from their families, instability, and receive less social support. In addition, children living in poverty generally experience less cognitive stimulation and enrichment in comparison to wealthier children. For example, low-income parents speak less often and in less sophisticated ways to their children, and are less likely to engage jointly with their children in literacy activities such as reading aloud or visiting the library, compared with middle-income parents. Low-income households also tend to have smaller designated play spaces for young children, have fewer home learning resources such as age-appropriate toys and books, and are less likely to have access to a home computer or the internet. For these reasons, it is not surprising that individuals raised in poor families have elevated rates of learning, behavioral, mental health, and physical health problems that persist into adulthood. Experimental manipulations of income among families, such as conditional cash transfer or welfare to work programs, are important approaches to study the effect of income on child development, as such programs often increase total income for families at or below the federal poverty line. Economists consider such approaches as one way to study causal effects of SES on child development. Recent work by Duncan, Morris, and Rodriguez, polling nearly 20,000 observations of children ages 2 to 15, found increased household income led to improvements in children's cognitive performance, specifically at younger ages. These investigators, along with others, found a $1,000 increase in family income raised children's cognitive outcomes, such as math and reading test scores, by 6% of a standard deviation. Similar effects have been noted in motivation, social behavior, and externalizing problems, with experimental increases in family income leading to better outcomes in these domains. Such results help to clarify that postnatal environmental experiences do contribute to some of the behavioral differences associated with poverty. Such effects have been found in the United States and other industrialized countries such as Brazil and Mexico. Preliminary evidence also indicates that such experimental poverty alleviation programs can positively affect neurobiology, with conditional cash transfer programs in Mexico found to be related to lower salivary cortisol in children. Thus, this body of work moves beyond correlational studies that have shown associations between social class and outcomes, and instead provides causal evidence linking increased income in poor families to improvements in outcomes in a number of domains. Some social scientists may see limitations in these data, as these social programs are not specifically designed to affect outcomes for children. Therefore, the exact mechanisms of any observed change are unclear. It is not known what lies behind the positive response to increased income or the negative effect of poverty on children. We still know very little about how impoverished environments lead to developmental problems. Studies in non-human animals where environmental conditions can be experimentally manipulated suggest candidate mechanisms for how environmental experience might affect central nervous system functioning. Conditions such as the variety and complexity of the stimuli in an animal's cage can influence different aspects of brain structure, including the number of neurons, glial cells, myelination, dendrites, synapses, and neurogenesis. Such environmental variables capture some aspects of extremely low-income home environments. Animals living in environments with lower amounts of cognitive stimulation or greater amounts of stress tend to have smaller brains and fewer cell bodies, dendrites, and synapses than animals reared in more normative environments. Remodeling of these cellular components, such as neural cell bodies, dendrites, and synapses, are theorized to underlie changes in gray matter, one type of brain tissue. The development of gray matter is especially important for understanding problems in cognition and behavioral regulation because this brain tissue contains neural cell bodies, dendrites, and synapses that support the processing of information and execution of actions. A large body of research has focused on these types of changes because learning is believed to be related to this kind of neural reorganization. Developmental cognitive neuroscience research in humans has found gray matter and also white matter, the other major type of tissue in the brain, are vulnerable to environmental perturbations. White matter is composed of myelinated fiber tracts and aids in helping distal portions of the brain work together. Important research in human twins examining genetic and environmental contributions of brain development, however, finds gray matter may be uniquely affected by the early environment and is less heritable than white matter. Research focused specifically on the neurocognitive effects of poverty helps to further clarify possible changes in the brain, with recent studies providing evidence of SES influences on executive function. The frontal lobe has been implicated in executive functions such as planning, impulse control, and control of attention, making it a candidate structure for investigation. The frontal lobe also has a protracted course of postnatal development and may be particularly vulnerable to the effects of early stress and experience. Additionally, alterations in the frontal lobe may be particularly important for the elevated rates of learning, behavioral, and health problems seen in children from low SES backgrounds. Exposure to poverty has been associated with decrements in attentional processes, working memory, and inhibitory control during infancy, childhood, and adolescence, and also in adulthood. The results of longitudinal research suggest that increased duration of a child's exposure to poverty is related to greater deficits in executive function and working memory in adulthood. The nascent body of research employing measures of neurobiology, such as electroencephalography, EEG, or MRI, also point to possible alterations in the frontal lobe being associated with poverty. In young children and adolescents, differences have been found in the resting frontal EEG, with lower activity being noted for children living in poverty. This result may reflect a maturational lag in frontal lobe development. Further work examining SES behavioral performance in the neural correlates of selective attention has found differences in evoked brain activity. Young children from lower SES backgrounds display lower electrical activity when deploying different aspects of selective attention, a cognitive process dependent on the frontal lobe. This video presents the findings from a study that examined changes in human brain structure from birth through the toddler years. Participants in this study ranged from 5 months to 4 years of age, covering a period during which there is a great deal of postnatal brain growth. Indeed, gray matter development accounts for most of the human brain's growth during the first few years of postnatal life. The researchers analyzed 203 MRI scans from 77 infants living in lower SES households, and also those living in more affluent households. Most of the infants were followed longitudinally, with the average first scan at age 13 months and subsequent scans approximately every half year until children were 4 years old. The infants in this study represent families with annual incomes that ranged from extreme poverty, less than $5,000 a year in income, to over $100,000 annually. The data suggests that low SES environments influence the rate of human infant brain development. Infants, toddlers, and preschoolers from lower income families began their lives with similar gray matter brain volumes, but had lower total gray matter compared with those from middle and high income households by toddlerhood. Differences in brain volumes between children from low and high SES households are not accounted for by infant birth weight, infants' early health, or differences in head size at birth. As infants aged and presumably had increased exposure to the effects of their environments, the differences in brain volume between poor children and those with greater resources widened. Smaller volumes in this brain tissue were related to greater behavioral problems in the preschool years. The differences seen were localized to the frontal and parietal volumes, with children from lower income families having smaller volumes in these brain regions. A large body of research has found the frontal lobe is centrally involved with executive functions such as planning, impulse control, and control of attention. Such differences fit well with previous research noting poorer executive function in children from lower SES backgrounds. The parietal lobe is important for sensory integration and aspects of visual attention. Development of the parietal lobe may be particularly important for connectivity between brain regions. These results extend a consistent literature in rodents, non-human primates, and humans, suggesting that early environments marked by stress or deprivation negatively influence brain development. These findings suggest that aspects of low SES environments have important functional implications for children's health and adaptation, perhaps by influencing key features of central nervous system development. In regards to neurobiological mechanisms, the differences in volume are likely due to neuronal remodeling rather than the birth of new neurons or neurogenesis. Volumetric differences associated with environmental experience are likely related to an increase in synapses, along with increases in supportive tissue, including both capillaries and glia. Studies with mice find changes in the hippocampus as measured by MRI to correlate strongly with axonal growth markers and not with measures of neuronal size or number, again pointing to remodeling of neuronal processes rather than neurogenesis. This sample was economically diverse. Children came from families with incomes significantly below the federal poverty line, FPL, as well as from families with incomes over 400% of the FPL. Specifically, children in this study were drawn from families with incomes ranging from barely 4% to over 400% of the FPL. Yet, these data cannot address issues of causation. This is because poverty carries multiple components of environmental risk. Other third variables, not measured in the sample, could lead to or be related to both lower family SES and differential rates of infant brain growth. Future research will be necessary to determine if one critical aspect of the environment is likely to influence children's brain development, or whether such effects reflect the influence of multiple factors in combination. Candidate factors might include the effects of household resources, environmental stimulation, crowding, exposure to pathogens and noise, parental stress, and nutrition. It is also possible that prenatal experiences affect brain development and reflect other disadvantages and risks related to poverty. Because humans are able to adapt to a range of environmental conditions, we must understand more about the level at which impoverished environments become toxic for children. Of important note, this dataset was designed to study normative development and screened out infants based on demographic, birth neonatal complications, and family psychiatric history criteria. This design may skew the sample because such issues are disproportionately represented among impoverished children. The present results, therefore, reflect so-called normal children living in lower SES. The results likely underrepresent the true effects of SES. Alternatively, one could argue that the exclusionary criteria may strengthen the implications of the results, as such factors as possible explanations of the association can largely be ruled out as factors lying behind the findings reported. Increased understanding about how environmental variations, such as socioeconomic disparities, affect human brain development and behavior has significant implications for advancing basic scientific questions, such as understanding genetic versus environmental contributions of brain and behavioral development. But even more important is that such understanding should lead to public policy initiatives directed at improving and decreasing disparities in human capital.